Good morning, everyone. It is 8:30. It's our seminar time to celebrate vision research. And today we have our friend from San Francisco, so not that far from here. And delighted to see you all here in the room as well on the Zoom. So with that, let's Hernan introduce our speaker today.
Hello and good morning. My name is Hernan Gomez, and I am a third-year graduate student under Dr. Pelcheski here at the Center for Translational Vision Research at UCI. It is my pleasure to introduce today's distinguished speaker, Dr. Jonathan C. Horton. Dr. Horton graduated from medical school from Harvard University, in addition to receiving his PhD in neurobiology also at Harvard. He completed residencies at Massachusetts General Hospital in Boston and Georgetown University Medical Center in Washington, D.C., in ophthalmology, neuro-ophthalmology, and pediatric ophthalmology. He completed a fellowship before establishing himself as a professor of ophthalmology, neurology, and pathology at UCSF. Since becoming a professor at UCSF since 2002, he has contributed to over 175 publications and received many awards, including the recipient of the Physician Scientist Award in Research to Prevent Blindness, the Khmer Prize for Excellence in Teaching, and the Bresser Prize in Vision Research. In addition to his many scientific achievements, he took part in the Arthur Jampolsky Lecture Series and serves as a review panel member of the Jules Stein Innovation Award in Research to Prevent Blindness. His early research sought to utilize the cytochrome oxidase method to explore a range of scientific issues, including the development of the visual cortex, the organization of V1 binocular border strips and monocular cortisones, and the effect of sensory deprivation and strabismus on cortical metabolic activity. Additionally, he sought to explain clinical disorders of vision through basic scientific research on the visual system derived from observations made in his own patients, resulting in a map of the visual field representation in the human primary visual cortex, which has become the definitive version adopted by textbooks. In recent years, he has provided a detailed quantitative description of the properties of intermittent exotropia in humans. Now, please join me in providing a warm welcome to Dr. Jonathan C. Horton for his talk, Exotropia, Physiology, Anatomy, and Clinical Management.
Thank you very much for those very kind words of introduction. It's a particular pleasure for me to talk as a participant in this UC Irvine lecture series because my son was an undergraduate at UC Irvine. So I spent quite a bit of time on the campus packing and unpacking boxes and lifting them into the back of my car. So the theme of this lecture series is translational research, and that is a theme that I've tried to strike in my research. I have a clinical interest in strabismus, amblyopia, and the care of children with misaligned eyes, but I'm acutely aware of the fact that the clinical management is often poorly grounded in basic science. It's empiric, and there are many fundamental problems related to the pathogenesis of strabismus that we still don't understand. So I have tried to model strabismus in monkeys. You see here a picture of one of my patients and one of my monkeys. We induce strabismus in these animals by cutting an extraocular muscle when they're very young, and then we're able to study the consequences of that manipulation as they grow up, and we're able to ask many questions in monkeys that can't be addressed directly in humans. So what I'm going to do in this talk is give you a flavor for the kind of approaches we've been using in the laboratory. And then midway through the talk, I'm going to segue into some very practical considerations for clinical management of strabismus and ways that we have improved the preoperative evaluation of children and the selection of candidates for surgery. So if we're going to discuss the problem of strabismus, of course, ultimately what we're concerned with is how the visual system generates binocular function and the maladies that afflict it. You know, of course, that in the lateral geniculate body, which is the thalamic relay station on the way to the cortex, the cells are segregated into monocular laminae. So there's no binocular integration really at the level of the thalamus. It is the neurons in the lateral geniculate body projecting to the cortex, and at the level of the cortex where binocular integration really occurs. So this is a view of the human primary visual cortex. It is the right cortex, and the calcarine cortex has been flattened here. So you're looking at it en face, as if you're looking down through the surface with ground penetrating radar so that you can see layer 4. Layer 4 is the layer in which principally the geniculate afferents segregate. This happens to be a specimen from one of my patients who succumbed to a malignancy, and about a year before he died, lost sight in one eye. I got the brain post-mortem, flattened it like this, stained each section for the metabolic enzyme cytochrome oxidase, and then used Photoshop to reconstruct layer 4. Now, you see a pattern here of light and dark bands. There are these dark regions here interrupted by these paler zones. These are the famous ocular dominance columns discovered by David Hubel and Torsten Wiesel. And what's going on is that the neurons lying here in the pale columns have lost their input because one eye turned blind before the patient died. So their physiological activity is reduced. That means that their metabolic demands are reduced and the levels of cytochrome oxidase diminish. So you see the contrast between the low levels of cytochrome oxidase in the column serving the blind eye and the robust normal levels in the column serving the intact 20/20 eye. I've superimposed upon this map of left and right eye inputs the coordinates of the visual field. So this is the central 1 degree. This would be the foveal representation. You can see that about half the surface area of the cortex is given over to the central 10 degrees of vision. And this is, of course, something that we all appreciate, that we have a very high concentration of cones and ganglion cells in the macula, very high acuity enjoyed in that portion of the retina. And so there's a lot of cortical territory devoted to the processing of that visual information. Now, there's a curious interruption here in the regular alternating mosaic of ocular dominance columns. This always looks to me a little bit like a whale breaching in Baja California out of the water. This is actually a zone that corresponds to the optic disc in the nasal retina of the contralateral eye. So there's a hole punched in the retina, punched in the wall of the eye to allow the ganglion cell fibers to exit. And in that zone, there are no photoreceptors. So there's a blind spot. And the temporal retina of the other eye fills that blind spot in. So knowing that information and knowing that this is the right striate cortex, the question is, which eye fell blind in my patient? And if you stop and think about it a little bit, you know that it must have been the left eye, because as I said, it's the temporal retina of the ipsilateral eye that fills this area in, and it shows robust cytochrome oxidase staining. So the ipsilateral right eye is intact and the left eye was blind. You can confirm that by looking out here in this region at the very anterior end of the calcarine cortex, where the ocular dominance columns peter out, and there's a small zone here that's lacking ocular dominance columns altogether. This corresponds to the extreme nasal retina of the contralateral eye, which represents the temporal monocular crescent. It's a monocular zone where the other eye isn't represented, and it's pale because the left eye was blind. Here's the matching cortex from the other side in that individual. So this is the side I just showed you, and this is the other hemisphere. The crucial point, of course, is that the contrast of the columns is opposite. So on this side, the blind spot representation is dark, and on this side, it's pale. And on this side, monocular crescent is pale, and on this side, the monocular crescent is dark. If something blinds or suppresses or injures one eye, or if you even suture closed one eye, you'll get an opposite pattern, yin and yang, of staining in the two hemispheres. And I emphasize that because later on in my talk, when I show you the left and right cortex from an exotropic monkey, you're going to see something very different. So try to hold this in the back of your mind for the duration of my talk, because I will come back to this principle. So you can see that in layer 4, the inputs from each eye are still segregated into these monocular ocular dominance columns. There's no integration at the level in the cortex where geniculate afferent synapse. Here's a schematic of that cutting a cross section through the cortex. You see a left eye ocular dominance column, a right eye column. If you put a microelectrode here and record from these neurons, their responses will be wholly monocular. The binocular neurons are generated through one or more downstream synapses onto cells in other layers, which blend inputs from the two eyes, and these binocular cells are sensitive to retinal disparity, so they encode binocular stereopsis. There are also cells in the deeper layers of the cortex, principally layer 5, that project back to the superior colliculus and other brainstem sites. These binocular neurons carry the descending message from the cortex to adjust the tone of the extraocular muscles to maintain the eyes in alignment. So without cortical visual feedback, it's impossible for the eyes to remain bifoveally fused. So if there's no binocular vision in the cortex, there's no potential to maintain binocular fusion of the eyes. So this is a critical feedback loop. Now, my PhD advisors, shown here around the time that I knew them and worked with them, David Hubel and Thorsten Wiesel, worked all this out in the 1960s and 1970s through a series of brilliant experiments. And this is one of the landmark papers in the history of visual physiology. I would say it is to visual physiology what the Canterbury Tales are to British literature or Western literature. And I urge you, if you have a rainy Sunday afternoon, to just pull this up on the web and sit down and read it. It's only 60 pages long. It'll just take you an hour or two, but it's one of the most beautifully written and fundamental papers in our field. Here, Hubel and Wiesel are recording from a single neuron in the cat primary visual cortex. And here you see above the visual field of the left eye, and here you see the visual field of the right eye. They're stimulating each monocularly using a slit oriented at the correct orientation that's moving back and forth. And here you see the spikes that are generated, and you can see spikes with two different morphologies, which they've labeled spike 1 and spike 2. So this is a binocular neuron because it spikes to stimulation of either receptive field monocularly. When they stimulate both receptive fields simultaneously, and you might ask, why aren't the receptive fields on top of each other? Well, the animal's been given a paralytic agent so his eyes can't move around, so that you can plot the receptive fields in a stationary location on the field. And whenever you give a paralytic agent to an animal, of course, there's also an anesthetic on board, the eyes will drift into an exotropic position. So the natural position of the eyes is exotropic. And you can see here that with simultaneous stimulation of the left eye's field and the right eye's field, not only do you get a much more robust response from neurons 1 and 2, but a third neuron pops in with a different morphology from neuron 1 and number 2. And this is an obligate binocular cell that only responds when both receptive fields are stimulated simultaneously and with the correct or optimal retinal disparity. So binocular stimulation drives neurons in the cortex more robustly and induces responses from disparity-tuned cells that encode depth. Now, the crucial experiment that Hubel and Wiesel did was to then go one step further and ask, what happens to these binocular neurons if we misalign the eyes early in life? So this is a slide that's right from their portfolio of old 35mm slides. And if you look carefully, you can even see some of the dust that clung to the plastic of the slide before I digitized it from their collection. This on the left is a profile of ocular dominance from a normal kitten raised with normal binocular vision. The y-axis is relative number of cells, and the x-axis is a reflection of the ocular dominance of the cell. If it responded equally to stimulation by the left eye or the right eye, it was rated a 4. And then if it favored one eye, it was labeled 1, 2, 3. If it was the other eye, it was 5, 6, 7. And you can see that virtually all the cells are binocular, with most preferring a fairly equal stimulation from the two eyes. These are litter mates that were raised with one eye muscle cut from a very early age, so the eyes diverged outwards and the animals were exotropic. Done at a very early age, this disrupts the binocularity of the cortex, and when you record later when the animals have grown up, you see that almost all the cells are monocular, preferring stimulation through either the left eye or the right eye, with very few that respond to both. So what's going on? Well, this was the simple hypothesis that Hubel and Wiesel put forward. And it's a very powerful idea that governs our thinking about this phenomenon to this very day. The idea is that that convergence on binocular cells that normally takes place, enabling binocular vision and a combination of the images from the two eyes, that's disrupted so that cells become either left eye cells or right eye cells through all layers of the cortex. And there are very few cells that are capable of merging the images from the two eyes and driving binocular function. And likewise, those descending layer 5 cells that normally would send a binocular input down to the brainstem break into left eye and right eye cells and are no longer able to mediate the fusion of the eyes on single targets. So this is the disastrous state of affairs that occurs when the eyes are misaligned early in life. And it's why pediatric ophthalmologists try to intervene so early to restore normal ocular alignment in children who demonstrate misalignment of the eyes. And if you don't restore that alignment during a critical period early in life, later on, when the child is adolescent or older, it's too late to restore this normal binocular wiring, and nothing can be done really to restore normal binocular vision. You can operate on the subject, you can reduce the deviation, you can improve how it looks cosmetically, but you never really establish bifoveal fusion. So I'm next going to introduce you to a form of strabismus that's very familiar to pediatric ophthalmologists. And I want you to forget about everything I've just told you, because all of these principles about a permanent breakdown of the wiring in the cortex generating binocular cells does not apply in this form of strabismus, because this is an intermittent form of strabismus. We call it intermittent exotropia, where mysteriously children or even young adults can alternate between bifoveal fusion and normal alignment and exotropia. So they're like a molecule that transitions back and forth between a cis and a trans isoform, and they do it seamlessly without experiencing diplopia, and almost always even without appreciating that they've lost fusion. And I'm going to just show you a videotape of one of these patients. These patients are absolutely familiar to everybody who practices pediatric ophthalmology.
Right. Bring that back together, huh? Cover the left eye. Take it away. Bring that in. Can you pull your eyes in there, Brooke? Look at the movie. You look at the movie. For a near stimulus, just a finger. There you go. Look at the finger.
Look at my finger.
There you go. Great. Now back at the movie. Good. You're on target.
So you saw the eye deviate outwards after the paddle was covered, which broke binocular fusion. Then she wasn't able to regain it until a near stimulus, in this case just a finger, was presented to her. That caused her to accommodate on the finger, that increased convergence tone, and she regained fusion and is back to a normal binocular state. When she was deviated, she wasn't the least bit perturbed, and she didn't appreciate diplopia. And that is fatal, because without the appreciation of diplopia, she has no motive to adjust the tone of our eye muscles to bring the eyes back into alignment. So the key to understanding how this intermittently deviated state is tolerated by the subject is to understand the mechanism of suppression. A simple-minded idea might be that the eye looking at the target is perceived and the eye that's deviated is suppressed. So you have complete monocular suppression of the eye that's deviated. And that, in fact, is the general concept that most pediatric ophthalmologists believe, but it's actually something that had never really been tested rigorously through simple psychophysical experiments. So we decided to map the perception mediated by each eye in subjects with intermittent exotropia, and we used a form of perimetry that's dicoptic. And what do I mean by the term dicoptic? I mean that one stimulates a subject under binocular conditions, but one has a way of tagging the stimulus in each eye so that you, the investigator, know which stimulus was presented to which eye. And one of the simplest and classic ways to do dicoptic visual stimulation is to use color filter glasses, which pass one wavelength to one eye and another to the other. And here's an example of how we do this dicoptic perimetry. Here's a subject, you're looking head on at her. She's wearing these red-blue goggles over the eyes. Here she's looking at this tangent screen. We present a stimulus for fixation in the center of the screen. This is a red stimulus, so she'll see it only via the right eye, and she'll fixate on it with the right eye. The background here is a blend of blue and red. It's purple, so that is perceived by both eyes. And then we present stimuli in the periphery, and we simply ask the individual to report to us the color of the peripheral stimulus. It's on for just a few hundred milliseconds, so there's not time for the subject to saccade out to that stimulus and see what color it is. And of course, we tell our subjects, keep your fixation on the cross, and we monitor the position of both eyes with an eye tracker. So if they cheat and try to look away, we discard that trial. So we do hundreds and hundreds and hundreds of trials. And this is an example of what the experiment is like for the patient with intermittent exotropia. There's your blank screen. Fixate with the right eye. Peripheral stimulus comes on. Subject should say blue. That'll be seen only via the left eye. Here's another trial. Fixate with the right eye. Subject has to report red. Now the subject will switch fixation to the left eye. Here, there's no stimulus, so the subject should say nothing. And these are woven into the trials just to catch the subject who occasionally may just make up answers or in a trigger-happy fashion report things that aren't there. Another trial with the left eye. Now, this is the important stimulus because it's purple, containing a mixture of red and blue. If the subject reports that this is blue, you know the right eye is suppressed over in this region of the visual scene, and vice versa. So these are the ones, these purple trials are the informative ones that tell us what portion of the visual scene is perceived by each eye. So here are some data from a typical subject, and this is the most complicated slide I have in my entire talk, but it's probably the most important, so let me take some time to go through it systematically. In the left column are all the fixations where there was a blue cross at the center, so the subject fixated at the center of the screen with the left eye, which is represented by this L. And these are all the red cross trials where the subject fixated in the center of the screen with the right eye. And you can see this is degrees along the screen, and this subject had roughly a 15-degree exotropia, whether she was fixating at the center with the left eye or with the right eye. These are the trials down here where the stimulus was blue. These are all the trials where the stimulation was red. And in these trials, there's no ambiguity about what the answer is. So these are kind of catch trials for reliability. And on 219 of these single color trials, she gave the correct answer 205 times. Why she got it wrong 14 times, I can't say. But these trials are occurring once every second or two. They're fast, they're quick, some of them are peripheral, some of them subjects just utter the wrong response. And you can see that here. Here's a red one, Here's a red one, shouldn't have been. The actual answers are denoted by the color inside each white circle, which is where the stimulus landed. And then the color in between is an interpolation. So we've, at rather low density, scattered the screen here, and she has reliably replied red for the red trials, blue for the blue trials. And notice that here, for example, where the red eyes deviated, she still has no trouble seeing a stimulus that's presented only to the right eye, a red stimulus. So the fact that the eyes deviated doesn't stop it from perceiving at all on these single color trials. Now, the purple trials are far more numerous because they're the ones that are informative about what the subject's seeing with each eye. And the subject is not aware that some of the trials are purple. So the subject is just aware that there are red and blue trials. What you see is that anything temporal to the fovea of the left eye is seen as blue. So the entire nasal retina of the left eye is perceived by the brain. And all the purple stimuli out here are seen as blue, and the right eye is suppressed. And likewise here, anything temporal to the fovea of the right eye is seen as red. So the whole nasal retina of the right eye is perceived. And in this area, the temporal retina of the left eye is suppressed. And then there's a zone where there's a transition between the positions of the foveas of the two eyes, where there is a trade-off in perception between one eye versus the other. And that zone here moves back and forth on the tangent screen with switches and fixation, but it doesn't move on the retina because its relative position between the two fixation points is fixed. You may wonder why there's a cluster of circles at every 5 degrees. That's because we do this repeatedly, 4 or 5 layers, to assess the consistency and reliability of the patient's responses. Altogether, a trial like this takes about an hour and a half. We take a couple of breaks in between to give the patient a chance to stretch and breathe. We've done this on about probably 25 patients now with very consistent findings. So here's a schematic of that result. This is the dicoptic perimetry in an exotropic patient of what portions of the visual field are perceived. And here's a projection of that onto the retinas of the two eyes. So gray represents suppressed retina. The nasal retina in each eye is perceived by the brain. The peripheral temporal retina is suppressed. And then there's a small zone in the temporal retina that is perceived. This sliver here corresponds to this sliver between the projection of the fixation point, the fovea, and the transition to the other eye. And this sliver here is this zone right here. So you can see at a glance that the larger the exotropia, the smaller the overlap in the visual fields, and the less peripheral retina needs to be suppressed until you get all the way out to a chameleon that has two completely independent eyes directed in different directions with no need for any suppression at all because there's no field overlap. And the largest extent of temporal field depression occurs in subjects who have the smallest deviation. The other remarkable feature here is, again, as I said before, that this doesn't move on the retinas as subjects alternate fixation. So here the subject's fixating with the right eye, here they're fixating with the left eye. This border moves on the visual scene, it doesn't move on the retinas. Now you can take this pattern of neural perception of the retinal input and predict what that will mean in the cortex in terms of metabolic activity. So if the peripheral ipsilateral temporal retina is suppressed in each eye, then out here in the peripheral cortex, remember, this is fovea, this is blind spot, this is monocular crescent. Here in the right cortex, the contralateral eye, its nasal retina will be dominant out here, and the ipsilateral eye's columns represented in gray here, will turn pale. And even the optic disc representation will be pale because it's normally served exclusively by the temporal retina of the ipsilateral eye, because this eye has a hole in it for the optic disc. So you'll have pale columns serving the ipsilateral temporal retina in each hemisphere, whereas in the central field representation, because you have input from each eye here and here, you'll have robust activity in the ocular dominance columns representing the central visual field. So we can test that hypothesis, not easily in humans, although I'm hoping one day my colleagues who work with fMRI will be up to it. But you can look in a monkey the same way we looked in some of those postmortem human specimens by raising an animal with exotropia. We do that simply by cutting the medial rectus muscle in each eye when the monkey's 4 weeks old, allowing the monkey to grow up, and then looking in the cortex. So here's a monkey. You can see that he's got a left exotropia. You can see that by looking at the corneal reflex of the ring flash that I used to take this picture. It's perfectly centered in the cornea of the right eye, so he was foveating with that eye. And then here it's displaced about 1 millimeter nasally, which signifies a rotation of the eye into an exotropic posture. That's about 7 degrees. That's the ratio for corneal displacement of the reflex and the magnitude of the strabismus angle. So here's the left cortex in that animal. And you don't see any striking pattern of columns here in the central visual field representation, but when you look out in the periphery, you can see some ocular dominance columns here. Note that their contrast is much lower than what I showed you in the human following complete blindness. In the case of complete blindness, there's not only loss of all sensory input, but even degeneration of the ganglion cells with transneuronal atrophy and a much more profound effect on levels of cytochrome oxidase. There's some spontaneous activity going on, so the contrast of the cytochrome staining is less, but what you can see is that there is a pale optic disc representation suggesting that the temporal left retina is suppressed. And here you see these ocular dominance columns, which I'll show you at higher power. I've in fact put an arrow on 3 dark columns. Now the big question then is, are these dark columns serving the right eye or are they serving the left eye? Well, the prediction is that the dark columns would be serving the right eye. To unequivocally establish to which eye those columns belong, I injected the right eye with radioactive proline, which is transneuronally transported up to the cortex to label the right eye's ocular dominance columns. So if I'm correct that these columns belong to the right eye because they're dark, the adjacent section, which was processed for autoradiography, should show silver grains corresponding to the injected right eye. And here is that matching twin section. And here is the label in the right eye's ocular dominance column. And you notice a complete absence of labeling in this blind spot representation because it's filled in by the ipsilateral left eye. So the prediction is borne out here. And when we go over to the other cortex, the right cortex, again, out here in the periphery, you see a robust pattern of ocular dominance columns, none of this ilk here centrally. There's the blind spot representation. And when you move to high power, and I now show you in the right cortex here, 3 typical pale columns marked with these arrows, those should belong to the right eye, since that's the eye that sends fibers from its temporal retina out to the right peripheral cortex. And indeed, if you look at the matching autoradiograph, these columns are labeled by that eye injection into the right eye. So this prediction from our psychophysical experiments in humans is borne out by our anatomical studies in monkeys that have the same condition. The remarkable thing here then, if I can just summarize this first portion of my talk, is that in intermittent exotropia, patients have this amazing capacity to flip back and forth between 2 states. So the fundamental wiring has to be intact because they're normal when they're fusing. And in a way, this is my favorite form of strabismus because it's the most salvageable. The visual system still works. And if you can just work a strategy out to prevent the lapses in fusion, you have a normal subject. Contrary to the popular belief that when one eye is deviated, it's completely suppressed. In fact, perception of the visual scene is shared between the two eyes. It's the peripheral temporal retina in each eye that's suppressed. You can see that when you look at metabolic activity in the cortex of monkeys. And remarkably, this means that the fovea in each eye is actually perceived centrally. You say, how is that possible? How can you have two 20/20 foveas being perceived simultaneously? Well, clearly the subject's attention is directed at one fovea that they're using to explore the visual scene. And all of these patients have a dominant eye that they prefer to use for fixation and they prefer to explore with. But if something comes up over on the other side that attracts their attention, they'll often alternate and put that fovea on it and use that fovea to guide and direct their visual exploratory behavior. And here's an example of that. This is a little bit of a kludge, but it's just to give you an intuitive sense of what's going on. Those of you who are ornithologists will recognize these as acorn woodpeckers. They're abundant in California. They're quite comical. One of the things they love to do in the summer is to form these granaries that they fill with acorns for the cold winter months. And you can find these pocked tree trunks that are filled with acorns out in the California forest. Well, imagine that you're an exotropic subject. You're looking at these two birds. You'll see this bird with your left eye. You'll see this bird with your right eye. And if you look at the scan path, as the right eye is looking at this bird, the left eye will sketch a fellow scan path on nothing of interest. The fovea will be here, and the fovea will be perceiving this portion of the visual scene, but there's nothing terribly interesting here that requires foveal quality vision, and the subject's attention will be directed to this bird. But then the subject might switch to be interested in this left bird. The fovea will begin exploring the bird's head here, and now the fellow slave eye will be scanning out here in the sky or on this blurry distant bough of a tree. Again, it's a fovea, but it's not using foveal quality vision to appreciate this portion of the visual scene. Okay. So I'm going to finish my remaining 10 or 15 minutes switching into more practical mode, addressing a question that is vexing for everyone in the field of pediatric ophthalmology, and that is you've got these patients with intermittent exotropia, and it's actually, I think, the most prevalent form of strabismus. As funding for grant applications becomes more desperate, the prevalence rates reported for strabismus have skyrocketed from initially 1% to 2%. Now I see figures that say 4 or 5%. I think overall in the country, they're still back around 1%. But out of those 1%, at least half of them are patients with intermittent exotropia. And the dilemma for the pediatric ophthalmologist is, how do we treat these patients? Do we send them off for vision therapy? Do we give them pencil push-ups to try to get them to accommodate and converge? Do we give them minus lenses in their eyeglasses to induce accommodations so that we increase their vergence tone? Or should we just operate on all these subjects? And why not just operate on them? Well, the biggest problem with operating on these subjects is you really haven't fixed the underlying brain problem. I mean, the focus on operating on eye muscles just misses the boat when it comes to strabismus, because fundamentally it's never about an eye muscle problem, or almost never. I have to say almost never in deference to Joe Deemer, but it's usually not an eye muscle problem. It's a neurological problem, and it's still there after you operate for intermittent exotropia. So often the strabismus recurs. I mean, the recurrence rates are just astronomically high. I mean, some people have quoted, 50% in 5 years. So it's a real dilemma, because you have to tell families, I could operate your child, but we may be right back in the same boat 5 years from now. So you have to select surgical candidates very, very carefully. And in order to facilitate that process, we've begun assessing how much of the time the eyes are actually deviated by using wearable eye tracking glasses to look at how frequently the eyes deviate out in a typical subject. So this is how the glasses work. It's a commercial product sold by the Tobii company. These are two miniature cameras that take a picture of the cornea. They look at the position of the pupil center, and then these are 8 illuminators which cast a constellation of reflections on the corneal surface. And the computer is able to infer, after calibration, from the relative position of these reflections in the center of the pupil, exactly where the subject is looking. These glasses are easily worn. They're tolerated by patients. They give robust recordings, and we're able to put these on kids as young as 2 or 3 years old as 90 years old, send people out for the day. And then at the end of the day, they send back the tracker to us and we can garner the data. I want to just begin by showing you just a little bit of normal data. We've been kind of fascinated by just how much normal people move their eyes in the orbit and what normal oculomotor behavior is. This is actually a recording from a normal subject. And normally I include the names of patients, but I didn't include the name of this patient because I have her permission to reveal her name. That's Jackie Duncan, my department chair. And I just want to make a plug for department chairs contributing to their faculty's research by engaging as test subjects. She wore the tracker for 10 hours. And this shows her vergence angle. Here are the positions of the right and left eye over that 10-hour period. And this is a plot of her vergence angle with time plotted on the y-axis. 0 degrees is fixating at infinity. And you can see she has a bimodal distribution of vergence angles, which is absolutely typical. This is time spent on her smartphone or on her computer. And this is time walking around and doing things at distance. We've recorded, at this point, probably 25 people, looked at population data, and everybody has this saddle notch for intermediate distances with a peak at distance and a peak at near. This is another interesting data point acquired in 18 subjects where we just looked at gaze angle of normal people engaged in their everyday behavior. So this is horizontal gaze, this is primary position, normalized time on the y-axis, and what you can see is 18 different individuals, each color-coded. And what's remarkable is that even though the eyes can rotate in the orbit horizontally, plus or minus 50 degrees, actually, subjects spend no more than about 1 or 2% of their time beyond 25 degrees from primary gaze. And the standard deviation here is about 8 degrees. So interestingly, we hate to move our eyes very far. We'd rather move our head. And that's actually pretty obvious if you ever go to Wimbledon or look at a tennis match and you look at the people watching the match, and they could just hold stock still and move their eyes back and forth. This is completely unnecessary behavior. But either because people hate that tugging feeling of moving the eyes, or maybe because there's a common motor command programming it, we tend, in fact, to do this, and you don't realize just how much you do it until you have a stiff neck and you can't. Okay, so here's how we test the kids with intermittent exotropia. This is a little girl with her dad. I've just put the tracker on her. They go home, and then at the end of 10 hours, the whole unit powers down. They put it back in this gun case and drop it off at a UPS store, and it gets shipped back to me, and we take the data off the SD card. Here's a brief movie just showing a typical subject. You see the left eye's position with the blue circle, the right eye with the red, and she's intermittently going to fuse, and you'll see a purple circle. So we can take that data. In this case, she wore the track for about 12 hours. These are breaks here. We take the patients, we tell them obviously take the tracker off when you're going to the bathroom. She wasn't going to the bathroom then, she was just washing her hands before dinner. And here you can see the vergence trace All of the positive values represent convergence. This is fixation affinity, and all of these downward spikes represent epochs of intermittent exotropia. So she had hundreds of episodes of intermittent exotropia over that 12-hour period. And here's the histogram showing what that looks like. Here is her distance peak. Here's her near peak. And this is her exotropia peak. Normally, individuals never have a negative divergence angle. So anything here to the left of 0 represents an abnormal finding. And in this case, the area under this curve amounted to about 25% of the time. It's 25% of the time, but you know, it's actually bigger than the peak for distance fixation. In other words, when she's looking off in the distance, more often than not, she's exotropic than fused. So the family was disturbed about this, and you know, they looked at this data, we talked about it, and they said, we want surgery. So I went ahead and did eye muscle surgery on her, straightened her eyes. And this is a recording that I obtained a few months later. It's a very brief recording. It just lasted an hour or two. I think I had her just walk around the hospital for an hour because I knew that I had aligned her nicely, and I just wanted to show that with this brief recording. Exotropia prevalence is 0%. Had her come back a year later. Family was very happy. They said everything's going great. I said, okay, let me put the tracker on her. I had her wear it for about 3 or 4 hours. And unfortunately, she's got an exotropia back here. It's smaller in magnitude than it was pre-op. Pre-op, it was 20 degrees. Now it's 10 degrees. That's the benefit of the surgery I did. And it's small enough the family didn't notice it. But this is the problem with intermittent exotropia. The deviation is back. And now I face the dilemma of, do I do another eye muscle surgery? And if I do another eye muscle surgery, is she going to be exotropic a third time? And I'll be faced with another operation. So for this, parents now have lost a bit of confidence in me, and they said, well, let's just wait. Let's do some more recordings. Let's see how she is. Let her get a little older, and we'll decide what to do next. Here's an example of another girl, had a very, very large right exotropia. I recorded her, and you can see that she was exotropic almost the entire time. This is her histogram. A huge peak out here for divergence. She has a very small peak here for distance fusion, and that's it. So this isn't a very hard case to decide. I'm worried that if this girl stays out there, she could lose the ability to flip back into moments of orthotropia. So I went ahead, I operated on her. Here she is 5 weeks after doing surgery on the medial recti in both eyes. I've also refracted her. I think it's very important to optimize acuity in both eyes because I think that a well-focused fovea acts like a glue to help hold the eyes together. And she's done very well. And here's her post-operative recording done sometime, I can't remember how long after surgery. Again, it's only 2 hours, but now she has a nice distance peak. She has a nice near peak, and that large exotropia peak is gone. And I'm continuing to follow her. In fact, I'm continuing to follow all of these patients. Whether I've operated on them or not, I'm following them serially, gathering data about the natural history of this condition and how patients fare after doing surgical correction. Here's a dilemma. Here's a 10-year-old girl. She's got an exotropia. I record her 23% of the time. There's her distance peak. There's her near peak. There's her exotropia peak. What do? Talk about it with the mom. This mom was very, very much against surgery. She was just that type of mom. I think her email address was something like herbgatherer@yahoo.com or something like that. So just surgery was not an option. So I said, okay, let's try some minus lenses. So we gave her some glasses with minus 2.5 sphere on top of her native prescription. And then I recorded her again wearing that prescription. And you can see that it shrunk her exotropia considerably from 23% to 5%, because when I put that minus 250 in her lenses, she then has to accommodate to overcome that, and that induces more convergence and reduces her deviation. And then on top of that, I gave her some base-in prism, and that eliminated her strabismus completely. So this is an option where you can titrate through judicious use of minus lenses and prisms and avoid surgery in select patients. I want to make just a few last points. I'm almost done. Prior to the advent of these mobile trackers, the way pediatric ophthalmologists decided whether patients should have surgery was to use something called a control score. And the control score, there are a number of different control scores, but the underlying principle behind them is that you break fusion with a paddle so that the eye becomes deviated behind the paddle. You take it away and you see how well the patients control that exotropia by bringing their eyes back in together. And do they leave the eye out for a long time, which denotes poor control, or do they bring the eye right back in, which shows great control? And I want to just make the point that that control score is very subjectively related to how often their exotropia is occurring. And here's an example of a patient in the clinic who has super good control of his exotropia, but that doesn't actually reflect how often his exotropias manifest. So I'm just going to show you a video demonstrating how well he can control his exotropia.
Byron, thank you for letting me take this brief movie. Just switch eyes to the other eye. Good, and now switch back. Excellent, and switch back again. And switch back. And now pull them together. Perfect, excellent. You're very good at that, Byron. Thanks a lot for letting me take the movie.
So you can see he's a gymnast. He can switch eyes on command. He can pull them together. He can let them go. You'd think he has great control. Shouldn't have a big problem. But there's his recording over 8 hours. He was exotropic 99% of the time. And this is actually his exotropia when he's fixating at distance. This is his exotropia when he's fixating at near. So when he's looking at something close up, his exotropia magnitude is smaller because he's exerting more convergence tone, but he's still exotropic. And that's actually his distance fusion, about 1% of the time. So I operated on him and was able to restore his normal alignment. And I think that this case is a warning that one shouldn't put too much confidence in these control scores as a way of monitoring the severity of exotropia. One thing I have learned in my interactions with patients, I always ask the parents, what percentage of the time do you estimate your child's eye is deviated? And then I actually go out and measure it. And that's what I've graphed here. The y-axis is what the eye tracker tells me. X-axis is what the parents tell me. And you can see that the majority of the points are below the unity line, signifying a higher estimate from the parents than the data provided by the tracker. And these green circles here are cases where the families reported, you know, that the eyes deviated 50% of the time, 40% of the time, 20% of the time, whatever. And the trackers actually measured 0% of the time. So if the family just sees it happen once in the day, they're often quite panicked about it, will report that the problem is quite severe, but it may just be a 30-second episode occurring once a day. And those are clearly patients in whom you don't need to intervene surgically. And I'm afraid that some of these patients may be getting surgery because of the vehemence of the patient's parents and the level of their concern. This is another interesting piece of information that's come out of these recordings. This is a plot of the magnitude of exotropia and how often it occurs. And there is a correlation. Those subjects who have larger exotropias are more likely to have that exotropia occur more frequently during the course of the day. So it's been a lot of fun using these eye tracking glasses. I get a lot of optometrists asking me if I'll record their patients for them. I have parents who I've recorded and they are back in touch with me wanting me to record them again. One of the embarrassing things that's happened, I only have one tracker, and sometimes I have two patients that come to clinic the same day, and I give the tracker out to the patient in the morning, and then the second patient comes in later on and I don't have the tracker, and I've got to have them come back another day when it's available. Families like this, and so do non-surgical eye care providers. I think there's a lot we can learn from this, and I hope that they'll become more popularly adopted in the field of pediatric ophthalmology. We could learn more about the natural history of intermittent exotropia, which is not well understood, The idea is that everybody with this condition tends to progress, but I think a lot of people realize that's not true. Vision therapy is very popular. A lot of optometrists recommend it. It'd be great to have more proof that it really works. Minus lenses are an effective alternative in some patients. There are pedic studies that show it works, and you can titrate the result by using these eye tracking glasses. You can use eye tracking glasses to help guide the decision about surgery. Be nice to set some threshold for what percentage of the time the eye is being deviated rises to the threshold of justifying surgery. And we still don't know what is the best operation. So I'd love to see a prospective study using eye tracking glasses to monitor the results after surgery so we can get some hard data about which operation works best and learn how often exotropia recurs after surgery. So I want to thank you for your patience in listening to this talk. And I also want to thank my collaborators over the years who've shared this work with me, Dr. Dan Adams, Dr. John Economides, Michaela Dilbeck, Thomas Gentry, and Justin Jeng. And I'd be happy to take any questions.
Hi, Dr. Horton. Thanks for your really interesting talk. Do you know anything about why these eyes will just sometimes go to the side and why it's not all the time?
There are factors associated with loss of fusion. People who have this condition report that when they're tired, it's more likely to occur. If they've had something to drink or smoke, it's more likely to occur. It's more likely to occur, I believe, during distance tasks than near tasks, although that's actually data we're sorting out right now, and that's sort of still in the field of anecdote. But I think what you're getting at is more fundamental, and that is, what is the trigger for the flip? And it's some kind of process where somehow the cortex drops the image in one eye, and I'm not sure what that trigger is, and I'm not sure where in the cortex suppression is mediated, and I've been unsuccessful in probing its basic physiology in V1, the primary visual cortex.
All right, let's go to Cher.
Hi, Jonathan. Thank you for the very interesting talk. I just had a few questions. In fact, maybe one or two. So first question, how many shades of blue and red could the patient report in your dichoptic perimetry test?
They just say blue or red. It's a super threshold target, easy to see. It pops out as red or blue, and they just simply give that answer, and we record it, and then later we analyze the verbal responses as a function of what the target was.
So I think you can already see where I'm going to. What if a person has a strong dominant eye, but not totally suppressed for the other eye? How do you get, how do you measure that? Yeah.
That all the patients have a strongly dominant eye. So an interesting thing about intermittent exotropia is that all intermittent exotropics tend to fixate preferentially with one eye. But under these testing conditions, there's no crosstalk between the filters with that noise background up there, and the stimulus is so bright and so easily seen against the background that it pops through and is consistently perceived in that dichoptic pattern that I illustrated, regardless of which eye is dominant in terms of oculomotor behavior and preferential use during free viewing.
Okay. Sorry, just one more question.
Let me add one more thing. There are the occasional patients with intermittent exotropia who deny that they have diplopia, and we stick them in our apparatus and we start the testing, and they say, wait a minute, I'm seeing red and blue. And they see both. They don't completely suppress. So they're like an adult who acquires ocular misalignment, has diplopia, and in such an apparatus, when presented with the eyes misaligned with purple stimuli, they'll see a red and a blue dot, and they'll be separated exactly by the deviation of the eyes. And we do occasionally have an intermittent exotrope who says that and says, I see double. And when that happens, we just throw up our hands and thank that patient for their willingness to participate in our research and escort them to the laboratory door. So
Okay,
there
great.
are some patients who just, despite reporting that they don't have diplopia, do become diplopic on this test, and we can't study their suppression because they're not suppressing when we engage in this test. Perhaps that's what you're driving at.
Right, exactly. Yeah. So my next question is, sorry, I'm taking too much time. How many % of straps are intermittent exos? The reason why I ask this question is because it seems like if you just cut the extraocular muscles in the monkey, you make them intermittent exotropia. But we know that there are also straps that really just, they're not alternators. They just suppress one eye totally.
So the monkey model is imperfect in that it's not intermittent exotropia, it's constant exotropia. So we don't have an intermittent exotropia model in monkeys, and the genesis of the strabismus is really different in the monkeys because it's totally mechanical it's more like that Hubel and Wiesel experiment that I illustrated early on. It's not the human state, and that's a flaw in the model and a limitation of its use, although there's still many ways that model is valuable. There are, as you well know, some patients who develop early intermittent exotropia. There's no treatment rendered, and they do become permanently exotropic. But I think that's a very small percentage of the population. And they're, of course, the most worrisome because they can lose the ability to fuse if it occurs very early in life. But in general, I think intermittent exotropes probably account for about 50% of patients with strabismus, and 95% of those do remain intermittent.
Okay, thank you.
All right, I think Vivek had his hand up first. Dr. Patel, are you there?
Yes, I'm here. Thank you. Thanks for the opportunity. Dr. Horton, I really appreciated that talk, and I learned something every time. I just had a question with respect to conditions like hemifield slide phenomenon and even other acquired forms of visual field loss like glaucoma in our adult patients. When these patients develop a strabismus, it seems that it's mostly due to that lack of peripheral vision, which then is not there to drive their fusion. Do you think there's something else going on there in these patients who presumably have a normal binocularity or stereopsis during development, and what makes it so difficult for us to bring these patients back into alignment? I'm wondering about fMRI studies that I'm interested in doing, where we might take a patient who has a bitemporal hemianopia, who then develops a exodeviation that maybe was intermittent initially, then becomes more manifest, and then after they have chiasmal decompression, improvement of visual fields, would that change anything at the cortical level? But what do you think? Is there anything that
You've asked a lot. You've asked such rich and interesting questions that if I were unleashed, I could spend about 20 minutes responding. So let me give very brief and incomplete answers to each question. First, there is a phenomenon of sensory exotropia, a completely different beast, where if an eye has low vision, the subject's not able to maintain fusion and the eye drifts out. And it's cosmetically unacceptable and very difficult to fix, because if you do surgery without a functioning retina, the brain can't fuse the images, the eye tends to drift out again. It's a very frustrating problem. That's a completely different phenomenon, of course, from the intermittent exotropia here, where everything's working, all the cells are fine, the cortex is fusing, and then suddenly fusion is lost. Now, in the case of hemianopia, you actually lose sort of half the glue. Half of the matching retinas homonymously are no longer able to contribute to cortical fusion. And there is a phenomenon associated with hemianopia, acquired hemianopia, particularly in children. A very high percentage of them develop exotropia, maybe 50% or more. And that's a fascinating phenomenon. And I have mapped the visual fields dichoptically in patients after the development of hemianopia and shown the suppression patterns they develop and so forth. That's a small paper we published a few years ago. And that's an interesting phenomenon, and brings up the question of the role of cortical drive in maintaining fusion. And finally, you mentioned the hemislide phenomenon, which I have in mind that being related to bitemporal hemianopia, often with chiasmal lesions, and that's a horse of another different color. And I think we got to talk about that offline.
No worries. Thank you. I appreciate it.
That's another very interesting rare phenomenon that, we'll leave that one.
Okay. Thanks so much.
All right, we have two Tonys with their hands up, but let's go with Dr. Moshan first.
Thank you. Hi, Jonathan. I enjoyed that. Thanks.
Sure.
I love exotropia as much as the next guy, but as you know, at least in animal studies, and I think also as my understanding in the clinical world, esotropia is often a very different beast. And I wondered how much of what you're talking about, particularly the patterns of suppression and so on that you see in the exotropes, replicates in some modified form in esotropes. And that's a question, I guess, which could be answered either with animal studies or with human studies.
Yeah. So esotropia is a completely different kettle of fish. And, exotropia is almost never amblyogenic. And I think one of the reasons is that the deviated fovea is not suppressed and the subjects alternate and it's intermittent. So, I get referrals for strabismus and I inquire, and if I hear that it's exotropia, it's okay, no rush. I can see your child in 3 months. If it's esotropia, it's much more urgent because esotropia is highly amblyogenic. And of course, in esotropia, the visual fields are more overlapped. So there's even greater need for suppression to avoid diplopia. Whereas in exotropia, you're unhinging the visual fields and there's less need for, at least regionally in terms of the visual fields, for suppression. I think a huge glaring hole in our entire field is the absence of high quality psychophysics in patients who have esotropia. It's a very difficult problem to study, particularly because it's usually confounded by the presence of amblyopia. But I have made some tentative forays in this area, but I'm not a card-carrying psychophysicist. And you have to, doing super threshold stimuli doesn't work. You have to actually measure thresholds, and it's a very, very difficult problem. And I'm not satisfied with the work that's been done in the field so far. So I think this is a very ripe area for exploitation and a completely different scenario than what I've described in my talk today.
Thanks.
Dr. Nesburn?
I'm really impressed with the science that you brought to this field. And I'm looking at what you have up on the screen, the wearable eye tracking glasses, and the different things that can be used for, perhaps. And that's very encouraging. My question is, what kind of adoption has there been in the community, in your community, to use this system, and how expensive is it?
Can I ask you a question first?
Sure.
Are there any other ophthalmologists in your family?
Yes.
Who?
My father was an ophthalmologist and my daughter's an ophthalmologist.
I think I know your daughter. She was a resident with us, right?
Absolutely, she was.
Would you give her a hug for me?
I will. She's terrific.
Okay. All right. I just didn't want to assume that she was the only Nesburn in the world. There are quite a number of different models of eye trackers that are available using different technologies, using AI, using different sorts of methodology and different prices. And there are a number of labs that are starting to purchase these trackers and use them in exotropic patients. Tana Roberts at Stanford is doing it, and I know of others that are interested in applying this. So I hope this catches on. It would be very gratifying to me. I think it would be great. And I would like to see widespread use of these trackers to evaluate these patients. In terms of what they cost, this tracker is very expensive. It costs $20,000. It was paid for by my NIH grant. although it was very expensive, computers were very expensive when they first came out. So were Texas Instrument calculators, and their costs fell rapidly. After I bought this, the company came back to me about 6 months later and said, "Would you like to buy another one?" And I said, "Well, maybe, but I don't have the money." And they said, "How about if we sell you one at half price?" So that gave me an idea just how squishy the profit margin was on the instrument. I didn't buy a second one at half price, by the way. I think the price will come down. They're very easy to use. And I think what'll really get ophthalmologists excited about using them is when they can bill for the use of them, and it becomes a clinical test that they can use in the office and get reimbursed for the time it takes to do it.
Right, right. Very good. Thank you. Excellent talk.
Dr. Brown?
Yeah, thank you so much. That was really, really thought-provoking talk. Kind of piggybacking off of the eye tracking technologies, have you looked at saccadic velocities and smooth pursuit and monocular situations? Is there an imbalance in the muscles in these intermittent exotropes?
What an interesting question. Yeah, we have looked at saccadic behavior. We've looked at target choice, and we've looked at the abducting versus the adducting saccades made by intermittent exotropes. And if you take a given saccade from primary gaze in the nasal versus temporal direction, they tend to make hypometric saccades more often than they do hypometric abducting saccades. So there does seem to be some deficiency in the adducting behavior, but whether that is muscular or brainstem or higher, we have no idea.
And do you know, do those hypometric or hypermetric saccades change after the size of the exotropia is modified with surgery?
I haven't studied anybody after surgery. I would think if you, for example, did a bilateral medial rectus resection and reduced the prevalence of intermittent exotropia from 90% to 1%, I would think that that behavior would be improved, but I haven't studied that in any postoperative subjects.
Thank you.
Hi, Dr. Horton. Thank you for your talk. I was wondering, if the exotropia isn't severe enough to justify surgery, how would you decide between using like minus lenses to correct it versus alternate patching?
I never do alternate patching, and I don't understand the logic behind alternate patching. So to me, alternate patching would just be the recipe for making things worse. And I know that quite a contingent of ophthalmologists uses alternate patching, and I've never understood why. And I'm very open to somebody convincing me that it's a good idea, but I don't see it. It seems to me that diminishes the binocular functioning of the visual system.
Hi, Dr. Horton. It's Philip Kaiser. Thank you for the engaging seminar. It was really fascinating. Two quick questions. The first, do you ever have cases where this intermittent exotropia spontaneously resolves and the patients are normal? If so, did you ever learn anything about, any type of, eye exercises that they learn to do to correct the problem? And then the second question is, with your wearable eye tracking glasses, I was interested in the discrepancy between what the parents report versus what the eye tracker says. Is there any effect of wearing the glasses on these patients, wanting to converge more frequently? Is there any concern about that? Thank you.
You mean that wearing the glasses itself could alter their behavior?
Correct.
Yeah, it's a great point. We're capturing a 6 or 10 hour glimpse of their daily behavior, and it might be that they do less outdoor activity, and the glasses inhibit their behavior, and they just sit in front of the TV and accommodate, and they have less strabismus or something. And so we don't always know that we're getting a representative cross-section of their normal behavior. we've recorded quite a number of people consistently multiple times, and we see variation in, from recording session to one time it'll be 22%, then it'll be 35%, then it'll go back to 25%, and it'll wander around. And we presume that that's to be expected because activities vary from recording to recording and patients' freshness and alertness and so forth. I don't really, I don't want to really, I want to keep equipoise about whether this condition tends to inexorably progress or whether in many people it remains completely stable, or even in some could it spontaneously improve. But I have to confess, I don't think it improves or goes away in hardly anybody. But I do think that there's an enormous fraction of patients in whom it remains stable indefinitely. And I think we as ophthalmologists have to be very careful about using the threat as a cudgel to our patients of, if I don't do surgery, this could get worse and worse, and then your child could lose all binocular vision, and then we can't get it back. I think that's a very dire sounding warning to share with parents, and it tends to push them into surgery very strongly, but I'm not sure it's very often true. And so I strongly caution against that warning, I think that it could be generally a more stable condition than we actually recognize.
Jonathan, again, thank you so much for joining us, inspiring so many, and hopefully we'll see you a week from now at 8:30 when we're celebrating vision research.
Thank you so much. And, I may not have answered all the questions. There may be some in the chat. Please, it's jonathan.horton@ucsf.edu, and I welcome any feedback or questions, and I will respond.